Well, I mean, it just automatically stopped. I didn't know if it was full or what.
Oh, I don't know. It shouldn't be, but who knows? Then if you follow the right all the way around to the back, it goes down the back of the heart between the two ventricles. That's the posterior interventricular artery. So they got some diagrams in the book. Now, with these coronary arteries and veins, they are present on the preserved hearts. They're all the same color. I don't ask them on the preserved heart. So what that means is 28 through 36 will be on the models only. That's the only thing that it won't be on the preserved parts. Now the veins, same thing. I've only got 3 coronary veins. What happens? The arteries take blood to the heart where the oxygen is used. Then that blood is collected in coronary veins that are going to return the blood to the circulation. There are 3 coronary veins. There's one, starts in the front, goes all the way up around the left side and into the right atria here. This one, it's a great big long one. It's called the great cardiac vein. Now the easiest way to look at the veins is look at it from the back. You got the great big one coming from the left side. Then there's this one in the middle in the back. That's a middle cardiac vein. It's in the middle. It's kind of nice. Then you see the small one going off to the right side. That's a small cardiac vein. Small, middle, and great. They all drain into a little bag-like piece of vein called the coronary sinus. And that coronary sinus then returns the blood to the right atria. Now the coronary sinus, you can find it on the models. You can find the coronary sinus. It's the only one of these I'll ask on the preserved hearts. The coronary sinus, I can go into the right atria and I'll see a little hole. You can kind of see the probe wiggling around right there. That's coronary sinus. So if you go into the right atria, you'll see a little hole at the base of it, and you can stick a probe in there and you're in the sinus. It's running right into coronary sulcus there. So you can find the sinus on the preserved hearts. None of the rest of the coronary arteries and veins will be on the preserved hearts. They'll all be on the models. Now, that being said, anything except the coronary arteries and veins will be on the preserved models. They could be on ththese models as well. I can ask anything on these, but specifically the coronary arteries and veins will be on the models. Now you might take a look at the models, maybe even initially, because they're easier to figure out what's going on, because they're all color-coded. You know, you can figure, okay, here's the right ventricle, right atria, left ventricle, left atria, here's the AV valves, you know, here and here. The artery leaving the right ventricle, this one here, pulmonary trunk. The artery leaving the left, this one here, it's the aorta. Now the blue and the red on the models, and that goes with all the torsos as well. What does the blue and the red mean?
Reds, arteries, and blues, veins.
Okay, good. That's the answer I was looking for, but it's wrong. I was walking down these paths to do that. Don't take it personally. But no, that's an easy mistake. Everyone does. Blue and red on these models does not mean artery and vein. An artery takes blood away from the heart under pressure. Veins bring it back under low pressure. These are both arteries. The red and the blue represents blood that is fully oxygenated or deoxygenated blood. Now here's what happens. Blood coming back from your body, your head, it's going to return to the right side of the heart through these 2 big veins. These are the superior and inferior vena cavas. They are veins. I'll show them to you in a preserved heart in a minute. They're very thin walled. They bring blood back to the right atria. Now that blood is deoxygenated. It's used. It's already been through your body. When it moves from the atria to the ventricle, has it picked up oxygen yet? No, it's still deoxygenated. And the right ventricle will pump it out the pulmonary trunk. It's still deoxygenated. That's why it's blue. The pulmonary trunk will then split into pulmonary arteries that go to the veins. That's where it picks up oxygen. It's now oxygenated. The model will be red. It comes back into pulmonary veins. See these little red things here and here? Into the left atrium. So basically everything connected to the right ventricle, what they call a pulmonary circuit, all the arteries are carrying deoxygenated blood. The veins are carrying oxygenated. So it's a backwards color scheme. Most of the blood vessels we can see, though, are not the pulmonary ones, or what they call the systemic blood vessels, which come out of the left ventricle, which is already oxygenated. That's been to the lungs. It goes out the aorta, which is oxygenated. And every other artery in the body will be red because it's oxygenated. Just be careful. Pulmonary and systemic circuits, the color is switched. Doesn't really mean artery and vein. All the veins in the pulmonary circuit will be shaded blue on the models. What else? All right. So we're going to have to do blood vessels starting next week. We do them all. All you have to do blood vessel-wise today are what they call the great vessels, ones going into and out of the heart. We've already done 2. Well, I've actually done them all. Pulmonary trunk leaves the right ventricle. Aorta leaves the left ventricle. So those are the 2 aortas. And then the sets of veins, you got 2 vena cavas going into the right atria. That's the inferior comes down from below, and the superior vena cavas. And then going into the left atria are the pulmonary veins. Now, you can see the pulmonary veins on the models. they're not on the preserved hearts. Because the lung and the heart are so close together, when they hack these things out of the carcasses, they usually cut off all those pulmonary veins. So you probably won't see them. Let me show them to you on these just because they're, one thing you don't get in the models that you get on the preserved ones is their thickness. All right, this thing right here, I'm in the right atria. There's two veins here. One has been cut in half, but this one's good. This is the inferior vena cava. Look at the wall thickness. I mean, again, it looks like a collapsed baggie. Without sticking my finger in it, you wouldn't even notice it's there. That is the vena cava. It's a big vein. Look how thin it is. So that's, I'm holding the right hand's the superior vena cava. That's the inferior. They go into the right atria there. Now look at this thing. That looks like someone's cut the heater hose on your car. What is that, a 1/4 inch thick, an 1/8 inch thick? That's an artery. They have a much thicker wall. Again, it's because of the pressure. You know, your aorta has 120 millimeters of mercury pressure in it. If it ruptured, you could be sitting in the emergency room in the hospital. You're still dead. These things evolved to tolerate the pressure. So they're very thick. So that's the aorta. How do you know it's the aorta, though? It's just all brown. You can stick your finger down in it and look, I come out in the left ventricle, so I know that's the aorta. The other one, kind of run along the side here. This one, it's still fairly thick walled. I put my finger in it. Look, I'm in the right ventricle. That's the pulmonary trunk. And you can see the semilunar valve right where my finger is too. So that's a pulmonary trunk. Again, you kind of got to, you don't have to use your fingers. You know, there's a dull probe in your boxes. If you're not sure, stick them in. And do be careful. Some of these are so handled by students, they have holes where there shouldn't be holes and things go funny places. But this is in the pulmonary trunk. And there we go. It's coming out right here. And that's in the right ventricle. And that's a semilunar valve, not an AV valve, so that's the pulmonary trunk. All right. Only other thing I'll mention, I think it's on this model only. 38 is on this model only. You won't find it easily on the preserved hearts, and I don't think it's on most of the other models as well. It's this little white thing you see right here. I'm mentioning this because we'll talk about it in lecture. This is a ligamentum arteriosum. This is just a ligament. It's not a big deal. What are we talking about? Because again, like that foraminal valve, in the fetus, there's a blood vessel here. It's called an arterial duct, which allows blood to go from the pulmonary trunk directly to the aorta. It's another lung bypass, pulmonary bypass. As soon as you're born, it just collapses and gets turned into a piece of connective tissue. That little white thing there is a ligamentum arteriosum, arterial ligament. That's only on this model. I don't think you can see it well on the other models. All right, what have I forgotten, Gail?
That's it.
There is a slide in here. I'm not going to be that worried about it. You looked at it last semester. You remember the slide of your, again, skeletal muscle, smooth muscle, cardiac muscle. What makes cardiac muscle unique? How'd you identify it?
It had a lot of nucleuses and also it's kind of segmented.
Yeah, it's, well, they're shorter, they're branching, they have fewer skeletal muscle, and the intercalated discs if you get a good slide. That's all that slide is. I'm going to say not worry about it too much. So that's your sheet of stuff. You got preserved parts here. You got trays. These large preserved hearts. Now these are nice to use because they're large and they're a mammalian heart. There's really no difference to speak of between a cow heart, a human heart, and a cat heart except for size. They have all the same parts in the same spot, same function. So I'll go over this heart, and I will tell you on lab exams, I prefer the preserved hearts to the models. There are a few things that I'll only ask on the models, but almost everything else I tend to prefer to ask it on the preserved hearts because, you know, they're real. So let me go through a few of these. All right, just orientation-wise.
Wait a minute.
You know, the heart would sit in your chest, something like this. The little pointy bit down here at the bottom, that's the apex, like the tip of a pyramid or something. That's called the apex. This part at the top where all the blood vessels go in and out, it's called the base. Seems a little backwards, the base is at the top, but that's it. Now, there's some layers associated with the heart. We'll get into a few of these in lecture on Monday, but surrounding all of your internal organs, all the cavities in your body, there's some things called serous membranes. As we'll see in lecture, these are double membranes that surround the cavity, and then they fold back and cover the individual organs. The space between the two membranes is filled with a lubricating fluid, because of course all these organs are sliding around. You don't want to generate a lot of friction, because then you get inflammation, and it will kind of be chronic, because it's not like the heart can stop and let the damage get repaired. So there are some membranes, however, some of them are not going to be easy to find in a preserved heart because they've been removed. Normally, and we'll see this in the cat, when we pull the cats out next week, I'll show you this. There's a loose-fitting bag around the heart. That one is called the parietal pericardium. That's number 5 on your sheet. That's one of the serous membranes around the heart. They call them the pericardial membrane. So the parietal pericardium is a loose-fitting sac. When they remove these hearts from the carcasses, it's usually cut off. Like on this one, you see this little kind of membrane. There's a bit of it right here. That's the parietal pericardium, just a piece of it. But it's like a loose bag surrounding the entire heart. We'll see that much better in the cats when we open them up next week. Now, A and B on there, you don't really need to worry about that in lab. I probably won't ask it. But these serous membranes in general are very thin. They're a layer of epithelium in the basal membrane, fairly delicate, with the exception of the parietal pericardium. This one is not delicate. I mean, I can hold up the whole heart with it. And we'll talk about this a lot in lecture, that's why it's on your lab sheet. But this has a fibrous layer on it as well. It's dense, fibrous connective tissue, makes it incredibly tough. So that's the two layers. You don't really have to identify them. The inner one is called the serous layer of the parietal pericardium, and this is a fibrous layer. And that has some functional significance for the heart we'll look at later. So again, not much of the parietal pericardium on the heart. But then the membrane folds back on itself and it covers the outside of the heart. So physically, what's attached to the outside of the heart, there's this thin little membrane. You can kind of pick a little bit of it off. There's not much to it. That is either called the visceral pericardium, or it's called the epicardium. Again, two names for the same structure. If you're talking about the membranes, you usually see this called the visceral pericardium. If you're talking about layers of the heart, they might call it the epicardium. So epicardium, visceral pericardium are the same thing. You can use either term, it's correct. Now, most of these hearts have been sectioned like so. So you can see the remaining layers. Again, the visceral pericardium or epicardium is only like a cell layer thick. It's not much. Then you see this massive amount of material kind of between my fingers. That's most of the volume of the wall here. That's all cardiac muscle, so that's called the myocardium. That's all the force and strength for pumping the blood. So almost all of this volume here, all of this is myocardium. Then the heart has some hollow cavities in it, chambers. These are lined with a material called endocardium. So again, endocardium lines the inner surfaces. It's, again, just a cell layer thick with a basal membrane. It lines the insides of the chambers, covers valves. All of the internal structures are covered with endocardium. Now, when you look at the heart, mammals have what's referred to as a four-chambered heart. That's not universal in animals. Fish only have two chambers, frogs have three, but the mammals and birds have four. Let me just do an ugly diagram of it. That looks something like this. That's my stick diagram of a heart. So you've got 1, 2, 3, 4. 4 hollow cavities, 4 chambers for the heart. Now, looking at this one, do you see this little bag-like thing at the top here? Fairly thin. That's an atrium. This is the right atrium. And then on the left side, you have the same thing. That's the left atrium. So the atria are very thin-walled, fairly weak chambers. They're just a little bag sitting on top of the heart. They are important for normal heart function, but they don't do much of the work. In fact, if your atria quit pumping right now, you'd probably be, in quotes, fine. You wouldn't be winning any marathons, but your heart would still pump, because most of the force generated by the heart are these things right here. These are the two ventricles. And if I open it up, you can see I'll put my right fingers in the cavity of the right ventricle, left fingers are in the left ventricle. The ventricles are the main pumps. If your ventricles quit, you just keel over dead almost immediately. They're the main pumps. The atrials, we'll see, are kind of priming pumps or top-off pumps. Their function's not as critical, but the ventricles are the main pumps. Now, you're going to have to be able to orient yourself on these hearts, because how do you know which is which? I got two ventricles, one's the left, one's the right, but which is it? Is this the left? Is this the left? You have to be able to orient yourself, or you'll get lots of stuff wrong. Now, the easiest way to figure out the left and the right side of the heart, most of them are cut like this. My fingers are in the two ventricles. My right hand is in the right, my left one's in the left. Now, you'll be able to see a difference between the left and right. So this is the right. What do you notice between the left and my left hand and the right? What's the difference?
Size.
The what?
The size is different.
Yeah, look at the wall thickness. This left ventricle, there's about an inch of muscle there. The right one, maybe a half inch. That is typical of all the mammal hearts you'll look at. Right ventricle is much thinner walled. It's not as strong. Another thing to note, if you touch the apex, and then I open it up, you'll notice the left ventricle forms the apex. The right ventricle just kind of looks like it's strapped onto the side. So that's the easiest way to orient yourself. Once you know left and right, okay, I'm good. This is the left. It's certainly more muscular, much bigger, forms the apex, left side. Then, what about the atria? Well, there is a hole, an opening, between the atria and the ventricle on one side. So if this is your right atria, right ventricle, there's a hole there you can put your finger through. I'll do that right now. Stick my finger through this hole. I'm in the right ventricle, so I know it's the right side. I stick my finger through, and look at that. I'm in the right atria. Why? Because it's connected to the right ventricle. On the left side, I can do the same thing. Go through that opening. Look at that. I'm in the left atria. So make sure you orient yourself. In fact, when you look at a mammal heart, it's really two pumps. You know, if you were really good with a razor blade, you could cut this thing right down the middle. And it is literally two pumps. You have a right heart and a left heart. They happen to develop in the same spot, but sort of mechanically, they need to be different places. It's two pumps, which is a little different than things like a fish that only have one. We'll talk about why we've got two later in the lecture. So make sure you orient yourself. Then, if you notice between my fingers, see that big sort of wall of material between my fingers? That's the wall of both ventricles. That's the interventricular septum. So there's a dividing plate. You do have to be careful on the lab exams. You know, let's say I did something like this. Let's stick a pin in here, and I ask you to identify that. What are you going to tell me? You could say it's the interventricular septum, right? Could you say it's myocardium? Yeah. How do you know what I'm asking? You have to look at the question. If I want the layer, myocardium, the question will say, identify, then in quotes, layer. If I want the structure, it'll say something like structure. And you can't really say that's the left or the right ventricle. Why? Because it's both. It's the interventricular septum. You've also got a septum. So interventricular septum is 14. 13 is interatrial septum. I can show that on the diagram easier. It's just the wall separating the left and right atria. You can see it on a preserved heart, but it's really hard because there's all this junk in the way. What I usually do is stick a finger in the right atria and a finger in the left atria. There's a wall of material keeping my fingers apart. That's the interatrial septum. You can't really see it. You kind of got to feel it. Now, there is one part of this interatrial septum I tend to ask, and that is the fossa ovalis, which is way down. Where's the fossa ovalis? Number 15. Let me just explain what it is real quick, and I'll show you kind of how to find it. Again, you really can feel it really easy. It's hard to see. I'll show it to you in the models as well. But the reason that part is on your sheet of terms because of what it does. See, if you look at an adult heart, you have two pumps. As we'll see, the right heart, right side of the heart, the right ventricle pumps blood to the lungs only and back. The left side of the heart pumps the blood everywhere else. It's called the pulmonary and systemic circuits. We'll detail in the lecture. But basically, both sides of the heart pump about 5 liters of blood per minute at rest. Now, that's fine if you're an adult. But if you're a fetus in the uterus, what are your lungs doing? Nothing. They don't work. You're not breathing air. You don't even ventilate. The lungs of the baby are collapsed. So is there any point to send all your blood to these lungs that aren't doing anything? No, it'd be very inefficient. So in a baby, there is a hole between the right and the left atria. It's called a foramen ovale, just an oval opening. What that allows for is in a fetus, in the uterus, blood that's in the right atria would normally in an adult, that all goes to the lungs. But now it can move directly from the right atria to the left. It bypasses the lungs. It's a way for a fetus to not waste energy pumping all this blood to the lungs. It just skips them. So it's a foramen ovale. But then at birth, you don't want to be skipping the lungs because the placenta isn't working anymore. So immediately when a baby's born, the first thing it does is inhale. And what that does is it changes some of the pressure relationships in the circulatory system. And there's a little flap-like valve here. It's kind of like a valve that closes once. When that baby first inhales, it's going to cause this valve to slam shut, and it grows closed. In an adult, then, you'll see this little thin spot here, and that is the fossa ovale. It's where the foramen shut down. And you know, if you have a baby, you hear like a reference of a baby with a hole in their heart, that's usually what's happening. There can be a defect in that closure of that foramen ovale, and they have a hole that shouldn't be there. They're pretty good at fixing that, you know, in utero now. They can go in surgically and repair the damage. So let me show you where that is in a model. It'll be easier for you to see. A model that actually shows it. So notice when you got the right and the left atria up here. If you look in the right atria, they'll show you a little thin spot in the back, and that's representing the fossa ovale. So you can kind of see it in the models. Well, it's way down in there. On the preserved hearts, what I usually do is get a pair of clamps, hemostats, and I clamp onto it with a label. So you have to follow them in. But basically, put a finger in the right atria and a finger in the left, and you'll feel almost like a saran wrap thin membrane. It's right between my thumbs. I know you can't see it, but you can feel it in there. It's very, very thin, flexible sort of membrane strung across that intraatrial septum. So that's the fossa ovale. Again, it's hard to see on some of these models. You have to kind of get up in there. You can see it, but I can't really show it to you well from way up here. That's your fossa ovale. Then, so we got the ventricles and the atria. Again, orient yourself by the size of the ventricle. Because again, on the preserved, they're all the same color. It's hard to tell. The models are going to be easier. You got all kinds of different colors. You can tell what you're looking at. This is a little trickier, but orient yourself by looking at the size of the ventricle. So you can always find the left. And then from there, you know, feel free. Even in an exam, stick your fingers through holes. Oh, left atria, left ventricle. There we go. That's the left atria. Now, some other things in the heart. The heart, it's a pulse pump. You know, if you cut someone's artery and hold it in the bucket, how does the blood come out? Does it consult like a garden hose? No, we've done that experiment much. But no, it comes out squirt, then nothing, squirt, then nothing. The heart, it's basically like a muscular Dixie cup or something. It has two stages. When it's resting, it's just sitting there like putting a cup under the faucet. Blood passively fills up this muscular cup. Then when it contracts, what they call systole, it squeezes that blood and squirts it out somewhere. So there, like any pump, there have to be various inlets and outlets to get the blood in and get the blood out. There also have to be valves. In the circulatory system, you want blood to move one direction. You don't want it to flow out of the heart, then back into the heart, out of the heart, back into the heart. Then it's just oscillating. It's not moving. You keel over dead. So in the circulatory system, you will find a series of what they call one-way valves. The heart has 4 of them. Now, all these one-way valves do is they allow blood to move one direction, but if blood tries to go the other direction, they will close. So you've got 4 you need to name in the heart. Get my scribble here. One of them, or one set, is located between the atria and the ventricles. Now, unfortunately, these valves have several different names. These can be called atrioventricular valves, or just abbreviated as AV valves. That's descriptive because of the valves between the atria and the ventricle. Now, what these valves do, they will allow blood to move from the atria to the ventricles. But if it tries to go backwards, they'll close. The analogy I use is, something like a door. Look at this door right here. This is just like a valve. Imagine there were air blowing from this room into this room. What's it going to do? The air would keep this door open, right? As long as the air is moving this way, doors open, doors open. What happens if the air changes direction and blows in? It's going to grab this door and it's slam it, right? Screen doors in the summer sometimes. That's where the valves work. They're completely passive. There really is a bit of connective tissue and epithelia, but it's like a door that's hinged right here. As long as blood is moving from the atria to the ventricle, it'll push the valves open. And the pieces of the valves are called cusps. If the blood tries to go backwards, basically the blood grabs these valves and it'll slam them shut. I will mention, you listen to somebody's heart, like just put your ear to their chest, you hear that thump, 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 thump. Normally blood moves through the heart in a streamline, it's laminar flow. Not a lot of turbulence, you can't hear it. But when the heart contracts, it tries to force this blood up this direction. When the valve shuts, that's the thumping you hear. That thump thump is caused by two sets of valves closing, and they vibrate the tissue and you can hear it. So that low dub or thump thump, the first heart sound is these AV valves slamming shut. So you got two, again, these two sides of the heart are very similar. You got one AV valve between the right atrium and right ventricle. You have a second AV valve between the left atrium and left ventricle. To separate them or distinguish them, they've got different names. The one on the left side is, you can call it the left atrioventricular valve, that's fine. This one you can call the right atrioventricular valve. But you may also see them called the bicuspid valve. On the left side, it's called the bicuspid valve. Basically, the cusps are like little doors. If you look at the preserved parts, the AV valve on the left side, the bicuspid valve, has just two cusps. There's just two flaps here, like two doors that open and close. That's why it's bicuspid, two cusps. You may also see this, sorry, this one's got three names, referred to as the mitral valve. Someone looked at it and said, you know, what hats like the bishop's mitre, it's a type of hat, kind of looks like that, I guess. And those that you probably hear mitral bicuspid valve more than you hear left atrioventricular valve. But all three of these are the same. You can use them interchangeably. Mitral valve, bicuspid valve, left atrioventricular or left AV valve, all referring to that valve on the left side between the atrium and the ventricle. On the right side, you can call it the right AV valve. It's also sometimes called the tricuspid valve. It's got three little flaps or doors. So again, let me pull up a preserved heart. How do you know what you're looking at here? You find the left side of the heart or the right side of the heart. Let's do the right, it's split over a little more. This one I can see easy. See this little flap? And I'll walk around with this in a minute. This is one of the cusps of the tricuspid valve. If I go through that opening, look at that, I'm in the right atrium. So that's the valve that's guarding that opening between the right ventricle and the right atrium. And that valve is called the tricuspid. Now, when you look at this, do you see those little strings on it? All right, those are only found on AV valves. What those are called, they're on your sheet. They're called chordae tendineae, which is number 24. Let me show you what they do. These valves, the AV valves, are very thin. They're not very strong. And the problem is when they slam shut, the ventricle is going to generate a tremendous amount of pressure. You get enough pressure that it'll basically blow the valve inside out like an umbrella in the wind. Blood goes the wrong way, you keel over dead. So to prevent that, there's almost like little guy wires. Those chordae tendineae are attached to the edge of the valve, and then they run down to the ventricle, and there's often a little finger-like projection of the ventricular muscle at the other end. And when the heart contracts, those chordae tendineae are kind of pulled down and prevents it from going inside out. That's what they're for mechanically. So the chordae tendineae are the little tendons, basically going from the ventricle to the cusp of the valve. These little bumps of muscle, the little, often very, finger-like looking projections of muscle, those are called the papillary muscles, which is also on your sheet. If you ever find a good one. This is a right AV valve. You'll see this one's very obvious. Big finger-like projection of muscle anchoring the chordae tendineae to the ventricle. So you got a papillary muscle, you got chordae tendineae, and you got the tricuspid valve. On the left side, very similar setup. You've got the cusp of the valve. This one's been cut right through it. You see all those chordae tendineae on it, and this is the left side because I can tell this is the left ventricle. So that's the bicuspid valve. So if it's got those chordae tendineae on it, it's an AV valve. None of the other valves have those. And just make sure you're on the left or the right side to know if it's tricuspid or bicuspid. One set of valves. You've got another set of valves. They are the 26 and 27. They're called semilunar valves. But blood comes into the ventricles through the atrium, through the opening guarded by these atrioventricular valves. When the heart contracts, the blood leaves through a different opening. each ventricle has basically two openings in it. An inlet that comes down from the atrium, and then it's got the outlets, which is connected to an artery. Arteries, by definition, are the blood vessels that take blood away from the heart. They'll be directly connected to the ventricles. So when the ventricle contracts, it's going to eject blood out of these arteries. On the right side, the arteries are called pulmonary trunks. On the left side, it's called the aorta. I'll show you those again in a second. But when the heart contracts, it squirts blood out of these arteries. But then when the heart relaxes, the blood's going to have a tendency, again, go backwards. It'll move from the arteries back into the ventricles, so you don't want it going that way. So there's another set of one-way valves located between the ventricle and the large artery leaving the ventricle. These are called semilunar valves because they're kind of made of three little crescent moon-shaped flaps. The one on the left side is called the aortic valve or the aortic semilunar valve. You'll often see it just called the aortic valve. And the one on the right side is called the pulmonary semilunar valve. Pulmonary, by the way, refers to the lungs. If something says pulmonary, it means it's going to be associated with lungs. Pulmonary trunk from the right ventricle only goes to the lungs. Now, what they look like on the preserved hearts, these are little, there's not as much to them. Let me stick a probe in here and see if I can catch a piece of one. Can you see that, Gail?
Mm-hmm.
That is the pulmonary semilunar valve, because this is the right ventricle, so I know it's the right side. And this is the semilunar valve leaving. See, there's a probe. That little thing is a cusp, which makes this artery the pulmonary trunk. So I'll move around now. See that? Not much to it. No chordate tendineae. That little, looks like a little baggy thing. That's the aortic, or sorry, the pulmonary semilunar valve right there. See, I'm in the pulmonary trunk. This is the right ventricle. That's it. That's one of the cusps of the semilunar valve, just right there. Just that little membrane-looking thing there. There's three of them. Three little sort of crescent moon-shaped cusps. That's the pulmonary, right there. Now it's the same on the left side. And I can put the probe under it. I'm in the left ventricle, and there's another semilunar valve right there. So if I go through that, I know I'm going to be coming out. That's aorta. Again, if you can find the valves and you know what ventricle you're in, you go out the artery, and if it's the artery attached to the left ventricle, it's aorta. If it's the one attached to the right, it's the pulmonary trunk. Let's see what else is on here. That's the semilunar valves. All right, now just some other landmarks. I always take things, flip them around, so there, I know this is the left, this is the right. This is kind of how it would be sitting in my body. You see this white line going right here? It's kind of lighter looking. All right, this is a groove. In the intact hearts, or the live preserved hearts, shouldn't say live, they're not live. These preserved hearts, the groove is filled with fat, but this line you see running right here, that is in the interventricular sulcus. The sulcus is just a groove. It's filled with fat, and honestly, that sulcus is running right where the septum is internally. So you see that line running around here, and then up the back. That's the interventricular sulcus. Okay, it's second to the brain. You have the sulci and the gyres. Sulci are grooves. So there's a groove between the two ventricles. So it's interventricular sulcus. Then there's another one. Again, it's just a landmark. This one's easier to find. There's a crack. I can stick my fingers in it between the atria and the ventricles, kind of running along the base here. That's the coronary sulcus. It's also where this big mass of white fat is here. And you can see that on the models as well. Let's see, the only other thing we need, got that. All right, the next batch, 28, 29, 30, 31, 32, 33, 34, 35, 36, and to some degree 37. There are blood vessels associated with the heart. They're called the coronary arteries and coronary veins. They're pretty critical because, you know, when you went over muscles last semester, skeletal muscles, can they work without oxygen? Yes. They can do glycolysis and fermentation, right? Make a lot of it, because when you're exercising maximally, they run out of oxygen. They will make ATP anaerobically. Cardiac muscle cells are unable to make ATP anaerobically, which means if you shut off the oxygen supply to a cardiac muscle cell, it uses up its ATP and it's dead in like 2 minutes. That's a heart attack. So it is critical that cardiac muscle cells get plenty of oxygen all the time. Now the problem you have, while it's true, there's blood where my fingers are inside this ventricle, which will have oxygen in it. Can that oxygen diffuse out to supply these cells? The answer is no. Oxygen moves by diffusion, but I don't know if you went over it last semester, diffusion is only effective over really short distances. If you move like 10 centimeters away from a blood vessel, you know how long it takes oxygen to get there by diffusion? It's something like 11 years. Terrible. So for any significant different distances, diffusion sucks, it's worthless. So even though you may have blood inside this ventricle, it is not going to allow oxygen to diffuse through this 1 inch of muscle at a sufficient rate, you'd be dead. So to prevent that, to get the oxygen closer to the cardiac muscles, they have their own blood supply. Let me pull out, I like this one to start with, Gail. There's a little branch of the circulatory system just supplying the cardiac muscle cells. So with this heart, and you can see this on many of the models, this one's just bigger, I can take the pulmonary trunk and the aorta here out. And what you see, that little red ring there, that is the aorta, just leaving the left ventricle. As it leaves, you'll see two little blood vessels going off to the left and the right, running along the heart here. Those are the coronary arteries. You start off with just two. The one on the left side, surprisingly enough, is called the left coronary artery. You gotta make sure you're oriented right. The one coming out of the right side is the right coronary artery. Okay, so no problem there. Now, of course, what happens, once the arteries leave, they begin to split. Every time they split, they change their name. So the left and right's pretty easy. Then you're just gonna have to memorize the rest. But if you look at the left coronary artery coming out, it goes for about 1 inch on this model, then you'll see it split. One branch goes down the front of the heart between the two ventricles. That is the anterior interventricular artery. It's kind of a descriptive name. It's running through the front and it's between the two ventricles. It's actually running in that interventricular sulcus. So that's the anterior interventricular artery. The other branch kind of circles around the left side of the heart. That one's the circumflex artery. And there's more, you just got a scattering of them. There's all kinds of names for these. Then, that's it for the left side. The right side, if you follow the right coronary artery out, when you get to kind of the edge or